We'll now dive into some data from actually this conference that dealt with SG. And maybe Karine, you can talk about the SG abstract.
Sure. There was a really interesting abstract talking about real-world toxicity of SG, and they looked at about 86 patients, and about 2/3 of patients had EV in the immediate prior line of therapy. A total of 71% of patients got EV total at some point in time before getting SG. And they found that the toxicity was very similar to what was reported in the trophy trials, so that general neutropenia, GI toxicity, and about 25% of patients had primary G-CSF prophylaxis, and none of those patients experienced severe neutropenia. So I think that does bring a case maybe to be considering putting G-CSF in those treatment plans as primary prophylaxis for those patients. Another interesting thing that I found with this study is that about half did not require dose reduction. So they stayed at that same 10 milligram per kilogram day 1 and day 8 dose for the duration of their sacituzumab treatment. So there's no efficacy data from this retrospective study, but it was interesting from a safety perspective to see how SG performs.
Yeah, absolutely. Very, very interesting data as well. Is this, well, you mentioned the G-CSF. Is this consistent with what everyone does, or?
Yeah, I'll tell you, I've run into insurance problems trying to give primary G-CSF prophylaxis, and I've really had to go to the mat with the insurance company to get them to approve it. As I recall, the numbers from the Trophy UO1 study was about 20% of patients had grade 3 neutropenia, maybe slightly higher. Neutropenic, febrile neutropenia rate was lower. It was on the order of maybe 10%, right around there. So to me, that's high enough to warrant giving primary G-CSF. So I think if you can give it, I think it makes a lot of sense. I think the downsides beyond the financial toxicity is pretty low, and preventing a neutropenic infection in a very advanced bladder cancer patient strikes me as a pretty worthwhile. Yeah. So I try and do it, and I think hopefully this data helps give more, support to that for providers who are looking to do this.
I totally agree with all of you. I use routinely G-CSF as primary prophylaxis. I think it's very important to reduce the chance of febrile neutropenia. I agree with you, Karine. This data that you presented, retrospective that you just discussed, will help that approach. I try to cite the ASCO guidelines and the patient demographics, more senior age of patients, multiple prior therapy sometimes.
Right. Prior radiation for some.
Prior radiation sometimes, comorbidities, performance status. So I try to get this up front, and I think this has helped me, to your point, preserve the dose. And we'll
keep people on track, keep people not missing doses. So
exactly, so
very helpful.
And the response rate in at least the Trophy U01 cohort 1, it was 28% as monotherapy. And the question is, what does this look like in the real cohort, in the real world? I think there was some data, Vadim, looking at that question from the United Registry.
Yeah, and that's actually one of the next abstracts we'll talk about. Of course, as SG is, I believe, a pretty potent drug, and we use it widely now, particularly in, certainly in the post-platinum, post-immunotherapy setting, but really we often use it in the post-Infortumab vedotin setting. And I think we'll consider using it going forward in the post now that we're in the EV Pembro age, so to say. But there was another abstract addressing specifically the question of Sacituzumab vedotin's, or Sacituzumab Govitecan's, I should say, activity in patients with variant histology. And this was, again, analysis of the UNITE study, which is a large retrospective study looking at outcomes in patients treated with targeted agents, patients with metastatic urothelial cancer, initially focused more on Infortumab, but now increasingly looking at other targeted drugs as well, like Sacituzumab. And so looking specifically at outcomes in patients treated with Sacituzumab, we see actually responses that are fairly consistent with what we see in SG clinical trials. So in the Trophy study, cohort 1, we saw response rates of around 27% in the post-platinum, post-IO setting. In this actually more heavily pretreated population, because most patients here in this retrospective analysis actually received Infortumab as well, the response rates were pretty consistent with that. It was around 23%. And even in some fairly aggressive variant histologies, we consider very to have very poor outcomes, like small cell neuroendocrine, we see occasional responses. So this is a small analysis, and so we need to sort of delve into this data more, and also probably in larger data sets. But I think it's compelling to an extent that we can give this drug in patients with variant histologies. And of course, I will, well, maybe Petros, I'll let you mention your trial, which is specifically of sacituzumab in patients with variant histologies.
Great discussion, and I can tell you that when we did the Trophy User 1 Cohort 1 study, we published the JCO paper that led to accelerate approval of SACI. We had about 10 patients who had enfortumab before getting SACI on that trial, and 3 out of 10 responded. So with small sample size, 30% response rate in par with a 28% response rate in the whole population, very small sample size. So the data you quoted that was from United study, I think it's relevant because it adds more real-world experience there. Now, at the University of Washington, we have this very interesting program, and we are very grateful to our patients who actually consent for a research autopsy, some of them. And they consent ahead of the time, of course, and we do some research to evaluate the targets like Nectin-4, TROP2. And we recently published a manuscript looking at the expression of TROP2 and Nectin-4 in those variant histology subtypes. And we actually saw significant degree of expression of the targets in histology subtypes, maybe the exception of neuroendocrine. So it's interesting to see that you saw some responses there. And we designed a prospective neoadjuvant trial for patients with histology subtypes, variant histologies, and if they have a urothelial plus a histology component, for cisplatin ineligible, not fit patients for cisplatin. Before radical cystectomy, we do Sacituzumab Govitecan monotherapy for those patients to look the activity in these variant histologies with a TROP2 targeting ADC.
Yeah, that's so exciting because then you get the tissue from the cystectomy and you can look at responders versus non-responders and do it maybe a deeper dive into the histology.
Exactly, biomarker work.
Yeah, it's interesting you talk about neuroendocrine because it seems Nectin-4 is a luminal marker. So we know that luminal tumors will have Nectin-4, and so the neuroendocrines may lose their luminalness or undergo EMT, whatever, and then may lose this. So I think we're still needing better therapies for the neuroendocrine-like or small cell tumors, which sort of by definition may not respond to a Nectin-4 agent. Interestingly, there was a DLL3 BITES presented yesterday that included some patients with small cell bladder cancer in which responses were seen there. And that was very small numbers, on the order of like 3 patients or so. But we need to see more. We need to do better for variants, especially for small cell.
Yeah, very challenging population to treat for sure, right? So it's great to see at least some responses in these newer targeted agents.